Hi guys, so today we are going to talk about DNA. So DNA stands for deoxyribo, that's the D, nucleic, and acid, A. So deoxyribonucleic acid is the molecule that holds our genes and gives us all the characteristics that we have. If you look down at the graphic below, you will see that it says on average, humans are 99% biochemically similar to each other. And actually, it's a little bit above 99%. So there's less than 1% difference in our DNA between individuals of the same species. And it's that little tiny bits of difference that makes us look different from one another. That's our variation within our population that we talked about with such a strength to us as a species. So what is DNA and what is it made out of? Well, first of all, we need to know a little bit about the structure of DNA before we figure out how it makes us what we are like. DNA comes in the shape of what's called a double helix. A helix is a spiral. If you've ever seen a spiral staircase, that's a single helix. DNA has two sides, and I'll show you a picture in a minute. It has two sides like a ladder, and then you take the ladder and you twist it. And that's your helix. And inside that structure, there's the monomers of DNA, and we talked about these also at the beginning of the year with organic compounds called nucleotides. Nucleotides, like these little things down here at the bottom, consist of a sugar, which is this little house-looking guy, a phosphate, which is the little blue ball up here, and a nitrogen base, which is this black box hanging off of the house. So how I like to think about this is I think about this as like the gingerbread house, like made out of sugar. Okay, and this is my garage with my vehicle in it that runs off nitrous. So that's my nitrogen base. And then I get a little weird with the phosphate, and I think it's like a giant ball of poop coming at the house. So poop for phosphate. So when we're looking at this DNA, we have these sugars in here, these deoxyribo sugars. These sugars, as you saw by the shape of the house, have 5 sides, which means they have 5 carbons on them. Off of these carbons come 2 different groups. The phosphate group that we saw as the giant ball of poo coming at your house. And then there's also, when there's no phosphate there, there's what's called a hydroxyl group, which is an OH group. And this is all chemistry stuff, which you guys have not had chemistry. But I just want you to be aware that there are different ends. The hydroxyl group will be on the 3 end, and the phosphate group will be at the 5 end. And I'm going to show you in a picture in a minute here. The sides or backbone of your DNA is made out of alternating sugars and phosphates. So if you look along your ladder, the sides of the ladder are all sugar and phosphate, and in the middle where the rungs of your ladder would be are where your nitrogen bases are. So to show you here, this is the 5N, 3N thing. So DNA, the 2 sides actually run in opposite directions. So see how the phosphates are over here kind of hanging off on the left-hand side? This is your 5N, so that's basically the 5th carbon that they're hanging off of. So they're all hanging off in the same direction. And if you look at the other one, they're hanging off in a different direction going this way, right? And so this particular end is missing its phosphate. That's where an OH group would be, and that's the 3N. And it's opposite from the 3 end over here. So they're always opposite of each other because your DNA runs in opposite ways, right? And you can see the S's for the sugars, and the A, T, G, and C's are your nitrogen bases. So your nucleotide is always 1 phosphate, 1 sugar, and 1 base. And that repeats over and over again, with the only variation being the kind of base that is on there. So what do those A's, T's, C's, and G's stand for? A stands for adenine, T for thymine, C for cytosine, and G for guanine. Okay, so you should know what they stand for, though most of the time we just use them as A's, T's, C's, and G's. And these guys pair up with each other in a very particular way. These are called complementary bases. It's like when you make a good couple, you want your couple to complement each other, like where you're bad at something, they're good at it, and vice versa. So these bases also complement each other and they fit together. So A goes with T, and the 2 lines here represent the number of bonds between them. And these bonds are hydrogen bonds, and you can see that right here, the little H's are for hydrogen. These are hydrogen bonds, and hydrogen bonds happen to be a very weak bond, and so you need that for your notes. Hydrogen bonds are weak. And there's a good reason why they're weak is because in order to make copies from our DNA, we have to be able to split it open. So we have to be able to break those bonds when we need it. So just based on the number of bonds, there's 2 bonds here, we must know that these 2 bases must be an A and a T. G and C have 3 bonds, and so this must be G and C over here. And so one of the ways you can remember which bases go with which is you can think there are apples in the tree and cars in the garage. Apples in the tree, cars in the garage, and that should help you to remember that. Now these bases come in different sizes depending on what they are, and they have fancy names. We have purine versus pyrimidine, okay? So a purine, it's the smaller word between the 2, so it's the bigger base, so they're opposite. Big word, smaller base. So the purines are A and G, all right? And if you look at them, they have 1, 2 carbon rings. 1, 2 carbon rings. So adenine and guanine are larger bases because they have 2 carbon rings. Cytosine and thymine are smaller. They only have 1 carbon ring. All right, so they are the smaller ones, so they're the big word, pyrimidines. And we learned on the previous slide that the apples always go in the tree and that the cars go in the garage. So A and T go together, 1 big one with 1 small one, and C and G go together, 1 big one with 1 small one. And what that does for us is keeps our DNA nice and straight. So here's a model where you can see the A goes with T, C goes with G, and then it's nice and straight. And this is that double helix shape we were talking about. That's that spiral shape. So how did we even figure all this out? It actually turns out DNA is pretty new science. It was not till the 1950s that we actually discovered the structure of DNA. But all this started with this Russian guy, Chargaff. Chargaff was born in 1905, and no, I don't expect you to remember that date. What I do want you to know, and what I want you to put in your notes, is this bolded statement right here. So Chargaff moved to the United States to do his research, and when he did that, what he discovered is there's always equal numbers of A's and T's and C's to G's. Well, this makes sense because we just found out that they go together. So it's like making couples at prom. If you're making boy-girl couples, you got to have 1 boy for every girl. So A and T's together, C's and G's together. So if you want 20 couples, you got to have 20 of each. So this was the first step to discovering the structure of DNA, is to figure out chemically that these must be the bases to go together because they always come in sets. So if I have 20 A's, I know I have 20 T's. But knowing how many of these I have, I could have any number of these too. I don't know, unless I know how many total bases, which we will practice later. Then we got Rosalind Franklin. Rosalind Franklin's kind of a sad story. As you can look at the years she was alive, she was fairly young when she died. She was only 38. And Rosalind Franklin was a woman with a PhD in the 1950s. In the 1950s, most women were still working in the home. So the fact that she was brilliant and had a high-profile job at a university, you can imagine that in the 1950s, many of her male coworkers frowned on that. She was working under this gentleman, Maurice Wilkins, and again, the only part you need for her is this bolded part. And she was amazing. She created her own equipment, built her own equipment so that she could take X-ray pictures of DNA. And it was these pictures that landed up helping other scientists to discover the actual structure of DNA. Unfortunately, because she was doing X-rays, and in the 1950s, they did not know that exposures to X-rays cause cancer. That Rosalind Franklin died from cancer from her work long before she could even get any recognition, even if she was going to. But it turns out her boss, Maurice Wilkins, took her work and passed it on to 2 other scientists, Watson and Crick, without her knowing, and taking full credit himself. So she never got any recognition for her work, even though obviously what she did was brilliant and really contributed to the process. Part of that was because she died before they could award anything to her, and part of that was just because she was a woman in a male-dominated position at that time. So this is Rosalind Franklin, and this is her famous X-ray pictures of DNA. Now, like this does not look much like DNA because the DNA was actually standing up and twisting. So this dark area is from the spinning of the phosphate and the sugar, and these are from the bases in the middle. And so because it wasn't straight across, but it made this pattern, that helped them to deduce that it was in a double helix shape, because if you spin something in a double helix, that's what you see. So Watson and Crick, however, they did a lot of work using her pictures and the information from Chargaff, because remember, scientists stand on the shoulders of other scientists. They worked together, a British guy and an American, and they created a model of the structure of DNA. And they were correct in their model. And so they got the Nobel Prize in 1953, along with Rosalind Franklin's boss, who did not actually do any of the work. So how does our DNA divide? We know that during cell division, specifically the S phase of interphase, that we have to make copies of our DNA before our cells divide. So we have all these proteins, enzymes in fact, and we know they're enzymes because they end in ASE, and that's what most enzyme names end in, that help this occur. And the enzyme name actually really makes sense. DNA polymerase. It's the enzyme that creates the DNA polymer, so that's what the name is. So DNA polymerase is the main enzyme. So 3 major events have to occur in order to copy your DNA. First, the 2 nucleotide chains, or the 2 sides of your DNA, have to separate, and that forms something called a replication fork. And you may have heard of like a fork in the road, where it splits in 2 ways and you have to decide which way to go. So it's going to split it open like that, and the enzyme that helps split it is called helicase. Helicase unzips your DNA across those weak hydrogen bonds. Then DNA polymerase will come in and attach, and they will pick up nucleotides that are floating around your cells from the food you eat. So you are literally made out of the food that you eat, okay? You're going to use nucleotides, the DNA from like the steak you ate. You're going to take that cow's DNA, break it down into the monomers, and reuse those to make new monomers for your new cells. So the new strand of DNA will hydrogen bond back together, and then what you'll land up with is 2 strands. Each of them are part old and part new. So here's a little diagram showing you this is the replication fork that occurs. So the blue strand is the old strand, and the green is the new strand. So when you see the green climbing up, and notice this one is going in one steady stream from the bottom up, and this one is actually attaching in fragments. So they're attaching in 2 different directions because DNA runs in 2 different directions. So how they attach is a little weird. But they will attach, and they will keep going until that strand is complete, and you'll land up with 2 what's called semi-conservative strands. So here's a little animation. That would be helicase. They would be breaking the hydrogen bonds. The replication fork is here. These are the new nucleotides attaching on either side by DNA polymerase, and that will continue the whole way down the strand until you have 2 strands, both half old and half new, or semi-conservative. Now, sometimes during this process, mistakes occur, all right? Just like when you're typing your paper, you have typos, right? So DNA polymerase not only adds nucleotides, but after it's done, it reattaches and runs through your DNA, checking for any mistakes. And if it sees a mistake, it will cut it out. So DNA polymerase is not just the writer of your genetic story, but also the editor of your genetic story. However, you have probably read a book at some point that still had mistakes in it, even though it goes through the writer and the editing process. So even when you have something checking the system, errors can occur, and this is where we get slight mutations. And these mutations can accumulate over our lifespan and cause abnormalities, which is part of the reason we have the aging process occurring, why your skin starts looking older and not as good as you age, because these little mutations accumulate. So the direction these things move in, DNA polymerase begins at many spots throughout your DNA in both directions simultaneously. So you can do this in a lot of different spots. Oh, that's the end for now, guys. We'll stop our video right there. So save your notes, put them in the Dropbox, and I'll see you next time.